Thank you very much for the kind introduction. My lab is primarily interested in the development of infectious disease vaccines using messenger RNA. And today I'm going to tell you about our universal influenza virus vaccine projects using modified RNA. Okay, good. So for all infectious disease vaccines, we use the nucleoside-modified and purified messenger RNA lipid nanoparticle vaccine platform. Here's a cartoon about it. So basically, the RNA is made using a linearized DNA template encoding the antigen of interest. The plasmid also encodes the regulatory elements of the messenger RNA, the 5' and the 3' untranslated regions, and the poly-A tail. Once the RNA is made, it undergoes a purification process to get rid of the double-stranded RNA and other contaminants that would be recognized as pathogen-associated molecular patterns by the immune system, and they would induce unwanted immune responses. And like I mentioned to you, the RNA is modified, so uridines are replaced with 1-methyl-pseudouridine that further increases protein production from the RNA and decreases innate immune activation. Once this purified and modified RNA is made, it gets formulated into lipid nanoparticles. It's needless to say that influenza virus infection causes significant morbidity and mortality every year. Currently used seasonal flu vaccines often provide suboptimal levels of protection. Most of these seasonal flu vaccines contain 3 or 4 inactivated viruses. If the vaccine strains match well with the circulating strains, then the effectivity of these vaccines can be 50 to 60%. But in some years, when the vaccine strains do not match well with the circulating strains, the effectivity can be as low as 20%. So obviously, there is a huge need for the development of more broadly protective vaccines. The messenger RNA lipid nanoparticle vaccine platform has many advantages. I think one of the most important advantages of this platform is the extreme, enormous flexibility that allows for the formulation of more than one antigen encoding messenger RNAs into the same lipid nanoparticle vaccine. And we definitely take advantage on this in this project and in many other vaccine projects. In our flu vaccine, we really wanted to target conserved antibody-accessible regions of influenza virus, such as the conserved stalk or stem domain of hemagglutinin, neuraminidase, and some part of the matrix protein 2. So this is an ongoing, very fruitful collaboration between my lab at Penn and several laboratories at the Icahn School of Medicine at Mount Sinai. The overall goal of this research project is to develop a vaccine that protects against influenza A and influenza B viruses, because they are responsible for human infections. And we broke this up into 4 different projects. We developed vaccines against protective vaccines against influenza A group 1 viruses, group 2 viruses, and influenza B viruses. And right now we are putting together the final formulation and testing it in several preclinical models. So today I'm going to tell you about the group 1 studies and the influenza B studies. So let's start with the group 1 studies. Like I told you, we came up with a combination vaccine. The HA construct that we used, it's called mini HA because it doesn't contain the head domain of the HA, it's only the stalk, because we really wanted to develop antibodies that are more broadly protective. Head-specific antibodies are also fantastic, but they are very, very strain-specific, and that's why many universal flu vaccine approaches uses different versions of HA constructs that target the stem domain. And the other three constructs were neuraminidase, matrix protein 2, and nucleoprotein from the A. Michigan 2015 strain. We decided to add the NP as well. It's an internal protein, but it's pretty well documented that it can induce very robust CD8 positive T cell responses that can potentially contribute to vaccine-induced protection. So we made 5 different vaccines. We made 4 monovalent vaccines containing the mini-HA, the neuraminidase, matrix protein 2, or nucleoprotein, and we made a combination vaccine containing all 4 components. The vaccination strategy was super simple. Animals received a single intradermal immunization with these vaccines, and then 4 weeks later, animals were bled and challenged with influenza virus, and then we looked at morbidity and mortality. One potential issue with these multivalent vaccines is that it's possible that you cannot induce a very strong immune response against each vaccine component. So to look at this, we immunized groups of mice with the 4 monovalent vaccines and the combination vaccine, and then looked at antigen-specific IgG titers by ELISA. And we were very, very happy to see that in most cases, we didn't see a significant decrease or any decrease in antibody titers. In the case of M2, we did see some decrease, but it was not that significant. So we wanted to, initially we wanted to challenge animals with the A. Michigan 2015 virus, but it's not lethal in mice, and we wanted to use a virus that kills the animals. So we decided to use IVR180. It's an H1N1 pandemic strain, and you can see at the bottom of the slide amino acid similarity in the vaccine antigens and the virus and the IVR180. So the HA and the M2 components were pretty different. NP, it's fairly conserved, and the NA was identical. So again, single immunization, 4 weeks later challenge with, on the left part, you see that initially we started with the normal challenge dose, 5 times the LD50, and it turned out that every single vaccine, the monovalent vaccines protect, and the combination vaccine also protects animals. And then we decided to use a very high challenge virus dose, 500 times LD50. In this case, only the NA vaccine was protective and the combination vaccine, but again, for NA, this was a homologous challenge because the vaccine antigen and the virus NA was basically identical. To look at the mechanism of protection, we did a serum transfer study. Animals were immunized 2 times, 4 weeks apart with the monovalent vaccines or the combination vaccine, and then 4 weeks after the second immunization, animals were sacrificed, and we collected serum from these mice and transferred this serum into naive mice that were then challenged with IVR180. And it turned out, not surprisingly, that the protection from viral infection was primarily mediated by antibodies. Animals that received the the serum with HA-specific antibodies, M2-specific, NA-specific antibodies, or serum obtained from animals that received the combination vaccine survived this challenge with any or no weight loss. It's not surprising that those animals that received the immune sera from NP-immunized mice didn't survive because, again, NP is an internal protein, and we do produce antibodies against NP, but they are not very protective. Next, we looked at the antibodies a little more carefully, and it turned out that only the neuraminidase component induced antibodies with, that neutralize the virus. It's not surprising because it's well documented that many store-specific antibodies act through Fc-mediated functions, and indeed it turned out that we could measure antibody-dependent cellular cytotoxicity activity in those serum samples that were obtained from animals that received the mini-HA, the neuraminidase, or the M2 vaccine. We looked at the cellular immune responses induced by the NA vaccine and the NP vaccine, and animals received a single immunization, and 12 days later we looked at CD4 and CD8 positive T-cell responses in splenocytes, and it turned out that both vaccines induced both CD4 and CD8 positive T-cell responses as measured by interferon gamma IL-2 and TNF-alpha producing cells. And in the G panel in the right bottom part, you can hopefully see that the NP vaccine induced a very robust activation of interferon gamma producing CD8 positive T cells. And we have not looked at the role of these T cells in protective responses, but I can easily imagine that they contribute to vaccine-induced protection. Next, we did a dose de-escalation study, and we determined the lowest protective dose induced by the NA endocombination vaccine, and we were actually pretty surprised to see that a single immunization with only 50 nanogram of NA or combination vaccine induced protection from IVR 180, so basically a heterologous virus challenge in these mice. And the most important data slide from this part is shown here. When we immunized mice with groups of mice with the 4 monovalent vaccines or the combination vaccine and the negative control Firefly luciferase encoding mRNA LNP, and then challenged these mice with different influenza A viruses, it turned out that the monovalent vaccines induced variable levels of protection. They protected against certain viruses but did not protect against others, and the combination vaccine induced protection from pretty much all viruses that we have tested in this study. So the conclusions from the first part is that modified mRNA LNP vaccination elicits strong antibody responses to multiple influenza antigens, and now it's not a big deal because companies and also many academic labs put together several or even many mRNAs in a single vaccine, but when we performed these studies, to my best knowledge, there was only one paper that showed that we can really put together several components and induce responses against each of them. And again, we were very pleased to see that this combination vaccine induced protection from a diverse panel of influenza A challenge strains. So let me tell you very briefly about the influenza B studies. They are very similar to these experiments. In this particular, one important difference is that when we started these studies, there was no HA stalk targeting influenza B construct available. So since influenza B viruses have two distinct lineages, the Yamagata and the Victoria lineage, we decided to use two full-length HAs, one from a Victoria strain and one from a Yamagata strain. And the NA, the NP, and the M2 were from a Victoria strain called B Colorado 2017. So we made these 5 monovalent vaccines, and we made a combined pentavalent vaccine that we tested in a low dose, 1 microgram from each component, and the high dose, 5 microgram from each component. We did a prime boost study, so we looked at the responses after the administration of an additional dose as well, and again we looked at antibody responses and also the breadth of protection induced by these vaccines. Again, similar to the group 1 studies, we found that when we put together 5 different mRNAs, we can induce robust immune responses against each of them as measured by antigen-specific as measured by ELISA. And not surprisingly, when the animals received the second vaccine dose, the antibody titers were significantly increased. We looked at the microneutralization titers, and in this case, we could measure microneutralization activity in not only against not only in anti-NA antibodies, but also in the case of the HA immunized animals. And the reason for that, because in this case we used full-length HA, so we have a lot of, we induced a lot of HA head-specific antibodies, and many of them are neutralizing. And we could also measure ADCC activity induced by actually any of these vaccines. Even in the case of NP and M2, after a boost, we could measure ADCC activity in the immune sera of these animals. I'm not sure if you can see these small plots perfectly. I apologize for that. So this is basically the challenge. These are the challenge experiments. Groups of mice were immunized with the monovalent vaccines or the pentavalent vaccine at a low dose or at a high dose. And then the groups of mice were challenged with either the ancestral influenza B strain, the B Li strain, or a bunch of Yamagata and Victoria strains. And the take-home is that similar to the group 1 studies, we found that the monovalent vaccines induced variable levels of protection. Sometimes against certain viruses they protected, against other viruses they did not protect well. But the pentavalent vaccine, the combination vaccine, even at a low dose, 1 microgram per component, induced protection against all of these challenge strains. We wanted to know if these vaccines can induce sterilizing protection. So animals were immunized again with the monovalent or the combination vaccine. 4 weeks, and 4 weeks later they were challenged either with a Victoria or a Yamagata strain, and 3 days and 6 days post-challenge, a bunch of mice were sacrificed, and we looked at viral titers in the lungs of these animals. And again, it turned out that only the combination vaccine, the pentavalent vaccine, induced sterilizing protection, even when we used it at a low dose. Again, single immunization. In the dose de-escalation study, we obtained similar results compared to the group 1 studies. As little as 50 nanogram combination vaccine induced protection from infection after administration of a single vaccine dose. And similarly, again, to the group 1 studies, the protection was primarily mediated by antibodies, and again, not surprisingly, primarily by anti-HA and anti-NA antibodies. So those antibodies that could neutralize the virus. So the conclusions are that most of the monovalent vaccines protected mice from infection after a single immunization, but when we used the pentavalent vaccines, there was no or just minimal weight loss, and we could induce sterilizing immunity. Very low dose, 50 nanogram. From the combination vaccine protected mice from influenza B infection, and the pentavalent vaccine was a broadly protective vaccine. Before I finish up, I just want to mention a very important limitation of this study. We used naive mice in all of these experiments, and it's very well known that in humans, pre-existing antibodies have a detrimental effect on vaccine-induced protective immune responses. So we started a bunch of studies with mice, ferrets, and also non-human primates with pre-established antibody titers, and now we are testing these vaccines in those animals to see if we can still induce protective immune responses. Again, the group 1, group 2, and influenza B stories are published, and right now we are working on the multivalent vaccine, again in mouse studies, ferret studies, and non-human primate studies as well. So finally, I'd like to acknowledge the people who contributed to these studies. Rafael Nachbagauer and Alec Frein spearheaded the Group 1 projects. They both joined Moderna, and actually Rafael is, not surprising. Rafael is leading the infectious disease vaccine program of Moderna. Megan McMahon did a lot of studies in the Influenza B project and also in the Group 2 project. And right now I'm primarily working with Florian Kramer and his lab. From my lab, Jean-Milly participated very significant in the Group 1 story, and Matt Pine, Hiromi Muramatsu, and Csaba Bajus contributed to the Influenza B studies, and the LMPs were provided by Acuitas Therapeutics. So with that, thank you very much for your attention.